Good afternoon, and thank you all for participating in today's Reef lecture, Cuba's Hidden Treasures, with Dr. Amy Apprill. I'm Beth Chafin, CCMI's Science Communications and Development Manager, and our lecturer today, as I said, is Dr. Amy Apprill. Dr. Apprill leads the Microbial Ecology for Ocean Conservation Research Lab at the Woods Hole Oceanographic Institution. Her research examines the contribution of microorganisms to the health and ecology of sensitive animals and ecosystems of the ocean. Dr. Apprill received a BA from the University of San Diego and MS and Ph.D. degrees from the University of Hawaii. While the lecture is ongoing, if you have any questions during the talk, go ahead and submit them in the chat box. I'll be moderating and I may jump in as Dr. Apprill is going and ask questions, and then we'll also have some time hopefully for questions at the end. I'm just gonna now hand over power to share her screen to Dr. Apprill. And
okay, do you see my screen now?
That looks great. I think we're
okay. Perfect. It's not the presenter mode, right? Just the regular full screen.
Full screen. We're good to go.
Perfect. Great. Well, thank you all for joining me today. I'm really excited to share some of the research that I've been up to, and I thank CCMI for the invitation to present to you today. I apologize I can't do it in person, and I know some of you were expecting to see me in the spring, so thanks for your patience, and now we finally have this talk going. So today I'm going to tell you a little bit about the research that my lab has been doing in Cuba. And Beth, if you want to take over the host again, we're getting some new admits.
Request a couple more people in. Thanks.
Perfect. Sounds good. So to get started today, I just want to talk a little bit about why coral reefs are so important. I know this is probably a review for many of you, but this is one of my favorite coral reefs off Micronesia. These are one of the most biodiverse ecosystems on the planet. They rival rainforests in biodiversity and are home to over 9 million species. Corals are animals with plants in their tissues, and they form the structural basis for this ecosystem, which has a variety of fish, invertebrate species living in all the nooks and crannies of the reefs. One of the things that I think about when I'm in this environment is just how does all this life survive? They're basically located in the deserts of the ocean, so that water is so clear because there's very few nutrients around. But the corals and their reef partners have just figured out a way to do it and to flourish in this environment. And that's what really makes coral reefs, to me, such a special place. So thinking about the Caribbean, this is a picture of Carey's Fort Reef from the Florida Keys back in 1975, and many of you are probably familiar with these Elkhorn corals. So this is what many of the Caribbean reefs used to look like in a variety of different ways, but this is just one really nice example of abundant coral biomass, a lot of fish life there as well. And over time, Carey's Fort and other reefs have experienced pretty significant devastation. So this was in 1975. And then if we fast forward to 1985, you see that most of those corals are indeed dead. You see that slime of algae and bacterial mats that are over the coral. And then if you move forward even another 15 years, 20 years to 2004, you see that it's just a rubble reef. And so even though those corals were once alive and vibrant, supporting an ecosystem, they're no longer there. And unfortunately, this is what we're seeing for a lot of the Caribbean reefs. I know in the Caymans, it's not like this everywhere, which is fantastic. But this is something as scientists that we're really concerned about and want to understand really what are those components of a healthy reef ecosystem. And so being a coral reef scientist in the 20th century, it's a 21st century, I guess, it's a little bit challenging because we don't have these really vibrant ecosystems to always go to and learn about. And so what I wanted to do, one of the first things I wanted to do when I came to to the Woods Hole Oceanographic to start my lab was to be able to go back in time and think about, well, how did these reefs work? How did they function? Especially some of these more invisible small members of the reefs, like the microbes. So how do you do that? How do you go back in time? Do you get a time machine? Just like Back to the Future and physically go back, which would be wonderful. Or do you try to find a place that might be a little bit more pristine than others? And so that's what drew me to Cuba. This is the island of Cuba. The Caymans, as you all know, are probably just right down here on this ridge. This is the largest island in the Caribbean, and it's also home to the largest island coral reef. So over 5,000 square kilometers of reef are there. Cuba, what's so special about it is it just has a really different history of land use compared to other Caribbean areas, and especially the Florida Keys, which I just showed you with Carrie's Fort there. It's had a fairly low population, and the population that does live there has not had access to all the coral reefs that surround it. So they just haven't overfished them like they have in other locations. And then the other neat thing about Cuba is that, especially their southern reefs, they've done a really nice job of protecting them. So they've been an elevated protection status, which many of the other Caribbean reefs didn't achieve until more recently, but they've been like that basically since the 1970s. And so that ends up being just a really nice protective ecosystem. And the other nice thing about those southern reefs is that they're just also very far from the land. So they sit down here on the southern side, tens of kilometers away from the main island, and many Cubans did not have access to those reefs. So there are a variety of reasons why it became so protected, and it's basically is indeed the most protected coral reefs in the Caribbean. So this seems like a really great place, right, to go back in time, start asking some of these questions about, how do these reefs really work? What are some of the mechanisms? What role do microbes play in them? But there's also a lot of reasons why it is not easy to work in Cuba. So why not Cuba? Well, as a US citizen, I can't use federal funds, research dollars, to go there and do my work because of the economic embargo that we've had against Cuba for a number of years. So that makes it challenging. And then the travel itself is tightly regulated. So, tourism, as we know it for other Caribbean locations, does not happen as easily. There's really regulated tourism, and some scientific exchanges can come, but they have to be really well planned out and documented. Communication is also very challenging there. I can't just pick up a phone and call a Cuban collaborator and start talking about a project together. We were really restricted to email alone. We certainly can't do Zooms or video Skypes just because of the internet situation in Cuba is not what we have here in the US. And there's also no FedEx, and this is actually really important for scientists because we have a lot of stuff and we need to get it to these places to be able to do our projects. So that, again, that makes it just another challenge. So there's lots of reasons why not to work in Cuba, and sort of one of the bigger ones too is that you can't just use a US credit card or a debit card to buy anything there or rent a research vessel. Like all that is not set up there. They're not set up to take the US cards. And so you really just have to come down with cash, and even that's limited on what you can bring. So there's a lot of reasons why it's not an easy idea, and that's why a lot of scientists haven't been able to do this. So how do you work in Cuba? So our team spent quite a bit of time trying to figure this out. The first thing we did was we developed a partnership. And so we got together with Cuban scientists from the University of Havana, very talented scientists, Patricia Gonzalez and Michael Armanteros. The Ocean Foundation was a great liaison for helping us figure out the logistics. And then I had a really determined collaborator that helped me put this plan together, and her name's Allison Santoro. And so what we did was basically spend a lot of time planning how we could work together, where would we want to go, what kinds of boats we could use. And some of this was in person just because of the challenges that I talked about with communication. So that was also a neat opportunity to go down and see this really special island. And we found that many of the challenging things that often I have difficulty finding on island nations such as the Caymans is like liquid nitrogen, and they have it there. So there's also some big pluses there. We use liquid nitrogen to freeze our samples. So a big learning curve we were on for many months to try to figure out how could we do this and what was possible. And we did. We figured it out, and we had some first naive adventures in Cuba that I'm going to tell you about first, and then I'll go on to talk about some of the more advanced adventures, sophisticated adventures, I guess, that we've had. So the first thing we had to do was show up with all of our stuff. And so this is what it looks like to have a team of US scientists arriving in Cuba to do some fairly sophisticated microbiology research. So we had about 30 pieces of luggage there amongst our team. So lots and lots of questions at the airport, but we ended up getting everything in no problem. So this included dive gear, but also scientific equipment, lots of vials and sampling containers, also pumps to process seawater. One of our collaborators was using a drill as well to drill down and get a paleo record of corals. So that's kind of what we look like going through the airport. We get a lot of questions about what we're doing. Our first adventure was on this La Reina vessel. This is a commercial dive vessel that's run by the Avalon outfit in southern Cuba. So what we're talking about here is here's the island of Cuba. And then this red square here has a bunch of dots. And those were the sites that we studied in this archipelago, which is called Gardens of the Queen, or Jardines de la Reina. And so these are the most protected reefs in the Caribbean as well as in Cuba. And we had 4 sites on the outer reefs and then 2 within the Gulf. Not only is fishing regulated, there's no fishing. Tourism was actually really tightly regulated in this region as well. There's only 1,000 diving tourists that are able to come and see these reefs each year. That's a little bit about where our research platform. This was our team. So on all these expeditions, we worked really hard to have a balance between US and Cuban researchers. We did not want to be the US scientists coming in to do everything. We really valued the collaboration that we received from the Cuban scientists who knew already a lot about the ecology of these reefs and some of the history as well. And so they were integral to this project, and especially site selection. So this diving boat certainly didn't have a lab like we do on our modern research vessels. And so the lab became basically any kind of spare space we could find. So up on the top deck, we would set up our pumps to filter all the water we needed to look at the microbial DNA. And so that's what that looks like. When we would process samples that we would gather from the reefs, we would do it at the dining room table. So this is MIT Woods Hole Oceanographic graduate student Laura Weber processing samples on the top deck of the boat here after her dives. And then indeed that dining room table would get converted into dinner just half an hour later. And so we were kind of like almost hot bunking the area. So lots of dual purposes. And then the food was absolutely amazing that we were eating there as well. So I felt very spoiled. And then occasionally we had to dip into the bathroom for additional space. So here we're actually ironically measuring some of the nitrogen compounds in the seawater using some of the additional space available in the bathrooms. So what did these reefs look like? Well, they were indeed special. So this is just an example of one of the reefs in the Gardens of the Queen. You see abundant fish and some of the larger reef building corals. And so here's one of our divers collecting transects of ecological data along the reef. We did find some reefs that were dominated by these Elkhorn corals that were just so special in many of the Caribbean reefs in the 1970s and early 80s. So these patches of reefs do still exist more in the higher wave zones. So that was just like a really memorable moment for me, swimming over these corals and feeling like I was back in my time machine. So that was our first cruise, and then we followed that up just 2 months later, hardly any time to rest, with another cruise into another location in southern Cuba called Armanteros, which is this box here in green, and it's magnified here. And with this cruise, we were able to have more sites. We went from just 6 to 16, so we doubled our capacity, collected a lot more samples. We were just more prepared this time. We kind of knew what we needed to work in Cuba. And we also abandoned the dive boat and went for the Cuban research vessel, which was just an amazing place to work, amazing platform. So this is a picture of the vessel that we worked on. This was a converted shrimping boat, which became the University of Havana's main marine science vessel, basically. And so it was very easy to work on, a lot of space. Again, we outfitted our team with about half US and half Cuban scientists. So here's all their smiling faces at the dock. And we had even more space to work on. So, so here was an area, this is one of the research assistants, Thomas, that's processing some samples. You can see that we actually have places to put our dive tanks, which is wonderful as well. So one of the things that was not quite as great on this vessel, and this is like what makes me sort of a hardy marine scientist, is that I was more excited about the space and the capacity for diving and the sort of independence from the dive industry, but we didn't really have bunks to sleep in. So we basically would bring our mattresses up to the top deck here at night, and everybody would carve out their own little space. And it was actually just a wonderful place to sleep and wake up to. Thankfully, no mosquitoes. And so one of the main research questions that we looked at on these two cruises are how are these reef microorganisms impacted by humans on these reefs? And so we took the data that we got from the Gardens of the Queen, which are these very pristine reefs, the Gulf of Batabano, or which I just finished talking about, which are more impacted. It's more overfished there, it's not quite as protected, and there's some resort development there as well. And then we also had the opportunity right after we finished our Gulf of Batabano cruise, we stopped by in the Florida Keys, and we did some of the same sampling that we did on the two Cuban sites. And so these are reefs that are overfished and near human development. So we ended up having this really nice gradient of human impact between these three reef sites. And so this turned out to be quite a bit of data. And Laura Weber, the student I showed you earlier, spent a lot of her dissertation working this up. And I'm just going to show you a snapshot of what I thought was like one of the more exciting findings that came out of this study. And I promise this is the only graph I have in the talk today. Otherwise, it's just pictures and stories. But here you're looking at these three sites, again, these three maps. And this graph is showing you the number of microbial species that we detected in the water. And we do this just by looking at their DNA. So we take a water sample, we collect all the microbial biomass, and then we sequence the DNA to find out how many microbial species are there. And what these are, these little boxes are, they're called box plots, and each dot is showing you a different sample point that we took. So in the Gardens of the Queen, which were more pristine, you could see that we have quite a lot of microbial species, from 300 to 400 there. And it's also, those boxes are pretty tight. And then we move over to Armanteros, as well as the Florida Keys, and the boxes become much, much longer. So there's a lot more variability in that data. And they also dip quite low in the number of microbial species. Now there's two boxes at each color because we're showing surface water in the lighter color and reefed up water in the darker water. And so what this data is really suggesting is that these Gardens of the Queen, these pristine reefs, have a really different microbial signature compared to the other reefs. And that we see this very high, consistent microbial diversity on these protected reefs. And this, to me, just like got me so excited, because this is a similar trend that we're seeing in a lot of other systems. So for example, the human gut. High microbial diversity equates to a healthier lifestyle, and lower microbial diversity often correlates with signs of disease or chronic immune problems. And the same kinds of trends have been seen when we compare geographic, or compare urbanized human settlements, I guess. So those of us living in more westernized, highly settled places actually have lower microbial diversity in our gut compared to some of the Aboriginal populations and communities in some of the less developed areas. And so this trend just mapped right on to what we know about microbial diversity in humans as well as some other animal systems, and it was really exciting. But for Gardens of the Queen, this was only 6 sites. So I also thought maybe we just didn't get a good sampling of what this environment looks like. And so, as soon as basically we finished those cruises in 2015, I started working really hard to get back to the Gardens of the Queen, because I knew this was a really special place that I wanted to learn more about. And so we went back after lots and lots of planning, and we went back in style. This is the Aleutian research vessel. It's definitely a premier research platform. It's outfitted for diving. They're able to fill tanks on board, which is something we didn't have with our other 2 vessels. Very comfortable beds. We were not sleeping on the deck anymore. And we also had a crew to really help us with all of the research. And so this was really just a wonderful opportunity that came about. This is a research vessel that's was provided by the Dalio Foundation, which is now OceanX, and they actually have an even better research, or more sophisticated, better equipped research vessel for the next phase of marine science that I'm really excited about. So this just provides us the opportunity to just do so much more science, which is exciting. But there were also some challenges. Like, there's a whole why not bring your own research vessel to Cuba list here. One of the first things we learned is the area we wanted to go actually prohibited outside vessels from coming in. So because that garden, one of the things that made Garden of the Queen so special was that it was highly protected, and outside vessels were not allowed there before, only the Cuban vessels. And so we had to get some special permits. We learned along the road that there were a lot of special regulations by the Cuban government, including who we could work with and who was coming on our boat. So normally as a scientist, you get to invite people to come on your boat, but I was told who was coming on my boat. Thankfully, they were great people, great scientists. And there were just a lot of unclear steps to gain the exceptions that we needed to the rules to be able to do this cruise. So we spent a lot of time on logistics, a lot more time than I normally spend, but it was well worth it. This is our team, again, of U.S. and Cuban scientists. So we were, again, half and half on this vessel. And this is a picture just outside downtown Havana. So those buildings that you see in the background are the old town of Havana, which is just a really special place. So just to remind you again where Gardens of the Queen is, here's a little video that's going to zoom right into that area. So this cruise, this is the entire archipelago of Jardines de la Reina, and we were able to make it to 30 total sites. And although the area is officially protected, there are some areas that are more protected than others, and those are in this central zone here. And that's mostly because that's where the diving outfit is, and it's just more regulated there.
Amy, are you able to just speak a little bit more about the different degrees of regulation? Stephen had a question about overfishing on the previous map, and he wondered if that's why boats are restricted and tourists as well.
Yeah, absolutely. So I mean, the protections are primarily on the fishing that goes there. So there is some catch and release fishing, sport fishing, but that's regulated. I forget how many people a year can do that. And then as you move outward on this archipelago, there is some government regulated fishing for certain species. And so that's why in the center, it's the most heavily and highly protected.
Okay, great. Thanks for that extra information.
Yeah. Great question. Thank you. So we spent about a month working in this area, and it was glorious. I had that wonderful research vessel to support our activities, and we had these beautiful reefs to visit. So I want to tell you a little bit about our science program that we pursued using this illustration. So we had divers out on all of these 30 reef sites collecting information about the benthic, life there, the health state of the corals. We also had divers collecting video transect data of the fish populations as well as sharks that were indeed present on these reefs. My group would go out with our fancy pumps and collect all the water that we needed to look at microorganisms, both in the water, but also associated with the corals. So corals have really important microbial symbiotic associations that help in their protection. Much of the Caribbean, including, I've heard Grand Cayman has some of this new emerging coral disease, stony coral tissue loss disease, which is indeed caused by a bacterial pathogen or group of pathogens. And so these samples that we were able to obtain from Cuba really provide us with a nice, pristine backdrop of what's normal microbial association or microbiome with these corals. So we were able to get a wonderful sample set of that. We had collaborators on board looking at the chemistry of the water, using some of the basic nutrient analyses, but also in more depth, looking at the entire metabolic spectrum of compounds present in those waters, or at least that we could extract and quantify or identify. We had one of our Cuban collaborators, Michael Armanteros, was painstakingly taking myofauna or sediment sand samples at each site, looking at some of the smaller microscopic life, such as the worms in these waters or in these sites, in these reefs, because they play a really important role in nutrient turnover in these reef ecosystems. And then this was a pretty special part of the project. We had an acoustic scientist on board as well, so measuring the sound or the soundscapes of the reef. And so, you know, just like there's a soundscape around all of you and even me right now, hopefully you can't hear all the sounds in my house, but these reefs emit a really unique soundscape that can tell us more about their health and biodiversity, as well as any human presence nearby too. It picks up boat noises very easily, as well as airplanes going over. And so we were able to do some soundscape measurements on these very special reefs. And so what we've been doing since this cruise in late 2017 is putting all these pieces together. And when you collect this much data and have so many pieces, we, you know, even now today, I don't have a full picture of what all this looks like, but I can tell you a little bit about some of the trends that we're getting, and they basically fall within this illustration here. So from the sites that are in the most protected definitely have the most fish abundance, as well as the diversity of fish that are present there. And the coral communities as well are quite abundant on those sites. And what's exciting that we've seen, we're still working on the microbial data, but we do see some consistent shifts between the more protected and the less protected reefs, including the more protected reefs have a lot of what we call oligotrophs. So these are microbes that are just really good at living in low nutrient waters, which is pretty consistent with a healthy coral reef. And the other neat thing we've been seeing from this data is that there's a certain populations of microbes that are really reduced that we would expect to see there. And so we think that there's more grazing on these microbes that might be happening by some of the corals, the sponges, some of the other life that's living there. And that's consistent with a hypothesis that's been tested in some of the Pacific reefs too. And so it's exciting that we're starting to see it here as well. Now within Jardines de la Reina, we don't have this full spectrum from a, you know, fully biomass rich pristine reef to the other side of my picture, which looks kind of like murky and modern day or 2004 Careysport Reef. But this just gives you an idea that indeed the reef ecosystem level changes that we can see with our naked eye are also being mirrored in some of the changes that we see with the microscopic data. And so that's really exciting that reef protection is important, and it does seem to make a difference for these reefs. A couple of other fun photos. We had some grouper that were pretty interested in helping us with the sampling. There's one trying to get at my students' Whirlpack bag there. Many of these reefs we encountered sharks, which was pretty exciting as well. We partnered with a couple media folks to document some of the cruise because it was such a novel undertaking. So Fusion Univision has a video. I was going to show it here, but I think we're kind of running low on time, so I can certainly share the link that could be passed on to all of you. And then I just wanted to make a couple other comments about this experience, and maybe if we have time at the end, I'll show that video. But one of the things that happened while we were out on that cruise was that President Trump clamped down on the Cuban travel and trade. And so this was pretty, we felt this pretty hard being out there, like living and working with Cuban scientists, just feeling like they were no different than us. You know, the main difference is that we had more sophisticated equipment, but, you know, they were equally intelligent, equally capable. It's just the resources in their lives were not of the quantity and variety that we had. And so we felt that pretty hard, and that here the U.S. was still taking this stand that they didn't want to economically trade and associate with Cuba, as had been happening under Obama. So that was sad for our group, but we endured, and we definitely ended the cruise on a really great note. In fact, this was Thanksgiving Day in 2017, which we came together in downtown Havana to give a joint U.S. Cuban overview of the cruise to anybody in the Cuban scientific community and government that was interested. And so that was really well attended. Here's my collaborator, Michael Armanteros, who's talking about an overview of the cruise. And just overall, like as a scientist, I feel so fortunate to be able to have done this work, because not only was it amazing and outstanding science, but it just gave me this perspective on a whole different culture that I've never had before, and such an appreciation for what the Cuban scientists have been able to do with such limited resources. And so, I mean, it made me feel very spoiled in what I have available to me, and made me appreciate just what they were able to do. I called them a society of engineers, because they were good at just taking what they had and making just, you know, equally good and relevant equipment and supplies that they could use for their science. So that was really neat to see, and something that I appreciated. And some acknowledgements for all the help that I received in this. There's a large source of funding, which came from Ray Dalio, the Dalio Foundation. They're now known as Ocean X, as well as support from Rob Meunier and Melissa Patrician, who really helped me spearhead the logistics. I had many people in my lab involved, and many collaborators as well along the way. So I'd be happy to take any questions, and if we don't, I have a few minutes to show you that video. So I'm very flexible here.
Thanks so much, Amy. This has been really interesting, and I think it gives cool insight not only into what is actually quite geographically really near to us in terms of reef ecosystems, but also we've heard a lot about the amazing reefs there. It's interesting to see what you've experienced, and also to look at some of the pictures of what your research expedition actually looks like. It's not always so glamorous, right?
That's so true.
We've got a couple of questions. Paul wants to know if you have any plans to go back.
Yeah, that's a great question. And I got that a lot right after I returned. And at that point, I was like, not for a little while, just because I was literally exhausted. I mean, I got off that cruise in 2017, and I was supposed to go right to the Virgin Islands, and that's after the hurricanes had just hit, Irma and Maria, and so we were supposed to do some rapid response work. And I was in a hotel room for like 3 days, just ill, because I was just so worn down from all, you know, everything that we did. And thankfully that happened at the end, and I recovered just fine, made it to the Virgin Islands. But that's just the level of like effort, I guess, that it takes, I think, to get these things done. So I'd love to go back. The things that I need are just funding now to take that next cruise. And now that I've been spoiled with a full-on research vessel, it's going to be hard to go back. But we have a better idea of how to do things. We have lots of follow-up questions. And one of the reasons I'm so excited about working on Little Cayman with CCMI is because it's just sharing, the same water as Gardens of the Queen too. So I suspect that some of these same trends that we're seeing in the gardens might also be apparent on the reefs of Little Cayman.
I know that we had another question about how you managed to establish that partnership with the Dalio Foundation and the Elucia. Can we go ahead and start down that path?
Sure, absolutely. So that really came about because my institution, Woods Hole Oceanographic, had a pretty established relationship with Ray Dalio and his vessel, the Elucia. So we were managing it for a number of years. And he's been instrumental to my career, actually, because I've gotten to go to Micronesia. I've gotten a couple trips through to Cuba, and then last year we did a full cruise throughout the Florida Keys to document the disease outbreak there too. And so, just really, the fact that my institution had this relationship, I was able to pitch this idea of Cuba. It was actually a round of proposals aimed at exploratory research. And, this is one area that, we don't really know these reefs that well. We know some basic information, but not some of the more sophisticated analyses that we were starting to do in my lab. So it was great. And now he's gone off and established Ocean X and working with the Ocean 5 donor group. And so, I think there's a lot of fun things that are going to be emerging in the future once we get through COVID for ocean science. So hopefully my research program will continue to benefit, but I suspect that we're going to be seeing just a broader range of marine science being supported by that new vessel that he's got.
Great. So we can see a question here in the chat from Jared that you mentioned Acropora seems to be now restricted to higher activity areas. Why do you think this is the case, and did you see that in Cuba?
Yeah, so Acropora palmata often likes these just higher wave energy areas. And so where we did see it in Cuba, indeed, shallower, higher wave impact, I think they like that nice clean ocean water coming in and flushing that area.
And we feed them for storm protection in those waves, right?
Absolutely, yeah. So those are critical species. They grow fast. They make these just, huge structures, so much habitat.
Mm-hmm.
And so thankfully the restoration efforts are starting to show that those can get replanted in some of these areas as well. Yeah, great question.
Could you just talk a little bit about when some of these travel restrictions ease, some of the work that you'll be doing in partnership with CCMI when you're able to come down here?
Oh, sure. Yeah, so I'm really excited about the project and working with CCMI. I've only seen pictures thus far, but but one of the things that we're trying to do is understand the role of the coral bacteria in helping them resist stress, especially heat stress. And so this is a big hypothesis in my field right now, is that certain bacteria might help these corals withstand some of the higher temperatures, that they have these protective features, whether it be actually, helping them scavenge some of the free radicals that are produced under these higher temperature scenarios by the coral and their symbionts, or providing more protection from opportunistic bacteria and pathogens, which might cause some of these demises that we're seeing. So it's being tested in a variety of ways. So we're going to do this at CCMI by running a temperature stress experiment on corals, but also transplanting corals in different environments and understanding the flexibility of their microbiome to change. And one of the exciting parts of the research is that in my lab right now, I have a student, very talented student, Lema. She's working on developing a more quantitative way to track the changes in the microbiome, which will allow us to more definitively say, this is happening or this is not, because the current methods don't allow us to really get a good handle on what does change look like by numbers. And then hopefully we'll go from there and do more projects together.
Yeah. We did have a question also from someone in the previous chat box. They were asking about if you have any sort of preliminary results that you can share from your time in Cuba.
Sure, yeah. And I just posted the link to that video too, which was,
Awesome. Thank you for putting
I
that
was
in.
going to show you like a 3-minute version, and that's actually the 20-minute, but I'm sure I can, I'm sure if you look around for that title, you can find the little preview one too. Yeah, so definitely within that, the archipelago of Jardines de la Reina, we found that in the more protected areas, that's where fish abundance and biomass was the highest, as well as richness, like the number of species that were there. And, you know, as a reef scientist, when I go down on reefs, I sort of pay attention to the corals because that's my primary study subject. But I would say that at these particular sites that were just so fish heavy, it was hard to pay attention to the corals because there were just so many fish around you and just so many distractions. And yeah, so that was a lot of fun. And then those most protected sites too, that's where we're finding these like funny signatures with the microbes. So those that are a little bit bigger and more nitrogen rich, there's a couple species in particular that we measured. They're not very abundant, and so that's what we think some of the life there on the reef is just eating them. And it makes sense, right? Like it's all a really tightly regulated system, and so we do think they're growing there, but they're just not as abundant as others. And then we're moving on to test some more hypotheses about the role of the microbial diversity in that I showed you before, but I don't quite have that whole bit of data yet to share. So stay tuned.
I'm just going to prompt everyone, if you have any more questions, go ahead and put them into the chat box. Let us know. I just wonder if you have any more insight. The stony coral tissue loss disease, or SCTLD, is becoming something that we're really very concerned about here, particularly because the DOE is trying to be so proactive about it on Grand Cayman. Do you have any insight into that? What do you feel like is the next challenge, the next way of maybe addressing or mitigating or combating stony coral tissue loss disease?
Yeah, I mean, it'd be nice if we treat it like we do COVID. I mean, that when we're in the marine environment, we just need to be really careful now if we interact with it or if we're in even proximity to it. So we know that it's water transmissible. We don't know really how long it can live in water, you know, to get to the next reef, but that does seem to be the primary transmission route, is through the water or through another organism that's in the water. And so rinsing your dive gear in a like dilute bleach solution is really what you need to do after diving. You know, if you can rinse your boat too after being in one of those areas. You know, you don't have to wear a mask. That's a good thing, but you have to be careful, yeah, about spreading it. And it's a challenge. Like, it's spreading really fast, and it's made it to a lot of places. We still don't understand much about what's causing it. Like, we know if you put a sick coral next to a healthy coral, the healthy coral is going to get it. Some species are more susceptible than others. We know that antibiotics, amoxicillin, can stop the spread, so that's why we think it's a bacterial pathogen. And our lab's been working on trying to ID the pathogens too. We definitely have some what we call indicator species, which might be associated with the disease or the pathogen. It's unfortunately in corals, like, we don't have the resources to even test pathogens like we do in other systems, at least as well. So things like cell lines, this is how it's done in humans and model organisms, and so you can just easily test things. So we don't have those kinds of resources. So it's almost like we have just not even the capacity that you do have to have for a lot of other diseases in humans or aquaculture and things like that. And it's just so important to the reefs, right? We're losing about a third of our coral species in the Caribbean right now just due to this disease.
Right. Regina wants to know, how do you give a coral an antibiotic?
Yeah, that's a good question. So basically what's being done is they drill like a firewall where the tissue or the disease healthy interfaces, and then fill that sort of area with an amoxicillin paste. So there's various kinds of paste treatments they've tried out. and basically it looks like just a filled hole in the coral, and it seems to work, that it does stop the disease. And then the other thing that's being done is in some areas, just whole healthy corals that are going to be really susceptible are just getting moved on land to like aquaria facilities until the disease sweeps through that reef, and then they can be replanted out.
Sure.
So those are the two, mitigation strategies I've seen.
Simon asked, so you mentioned that there was a lot of Elkhorn coral in the gardens. Was there a lot of Staghorn as well?
There wasn't as much Staghorn, yes. So that was harder to find. There was a lot of the Orbiceller Montastria corals, like the really large reef building. A lot of the pillar corals, the Diplogyra, which are, those are one of my favorites too. And I think they're gone now from the Florida Keys. It's like really sad because that one's so susceptible to the disease. But yeah, Staghorn not as prevalent.
Okay. Well, I think we'll wrap it up now. We've kept you for a little longer than we intended to anyway. So thank you so much for the lecture today. We appreciate your time, and hopefully we'll be able to have you down live and in person at some point in the not too distant future. And I just want to say a big thank you to all of our attendees as well. We will post the video from this recording on our YouTube channel, and we'll share it on social media once we're all squared away with that. And we do have one more in our Reef lecture series that will take place later on this year. So everyone that has attended, please just stay tuned. That will be presented, I believe, by our Director of Research, Dr. Gretchen Goodbody-Gringley. So one more before the year is through. Thank you again so much for your time, Amy, and hopefully we'll see you soon.